Welcome to Uppsala University and to UpTalk Weekly. My name is Lina Source Emilsson. I'm your host today at this popular scientific seminar series organized by the Faculty of Science and Technology every Tuesday. UpTalk Weekly is created to give alumni and everyone interested an opportunity to learn more about the research being conducted at our university and to meet and ask questions to our scientists. Your questions can be asked in the chat today, and I will bring as many as time allows to our guest in the end of the session. Our guest today is Sophie Sanchez, researcher at Uppsala University and a paleontologist that uses one of the most powerful X-rays in the world to invest fossil skeletons, to understand how the first animal with 4 legs could emerge from the water. And this is long before the dinosaurs appeared, and how they could adapt to the life on land. Welcome to UpTalk Weekly, Sophie.
Thank you very much.
Happy to have you here, and I would like to ask you to start up to tell us a little bit about yourself. Who are you and what is your professional background?
Yeah, so I'm Sophie Sanchez from the Program of Evolution and Development at the Evolutionary Biology Center. And so I'm a paleontologist before being a biologist, actually. So I'm studying fossils, which are, as you said, much older than actual dinosaurs. And I did my background, so I did my studies at the university in Paris, and I did my PhD at the museum in Paris. And then I came here as a postdoc and I decided to stay. So I got a permanent position and I decided to stay here and be a senior lecturer now at Uppsala University. And it actually all started when I was kind of little, this passion for paleontology. So when I was 9 years old, I went to an exhibition on dinosaurs. So I wanted to be, at that time, I wanted to be a doctor, like, you know, this actual medical doctor. And I thought I was a bit afraid of needles. So I thought, okay, if I can find like another way of approaching medicine, that would be nice. And I found at this exhibition that you could study the pathology of dinosaurs. So I decided that I would probably be the doctor of a dinosaur rather than the doctor of a human instead. And so this is how I ended up in paleontology and studying the bone tissue.
So what kind of bones or what kind of fossils are you working on? And your first project you worked on, what was that?
Yeah, so I'm working today, I'm actually working on fossils which are from the Devonian to the Triassic. So when I speak of the Devonian, it's roughly 380 million years old animals. And up to the Triassic, which is about 200 million years old. So I'm working on this large range of million years. And these fossils, they come from all over the world. So they come from expeditions that were led in the 1930s in Greenland, for example. But I also work on fossils which are from Canada as well, Arctic Canada, and fossils which are from Europe as well. So it's a large span of fossils and type of varieties of locations. And the first project I worked on was on a small fossil from Czech Republic. And I here have a small humerus of this fossil. So this is the tube in which you have just the humerus, which is here. So this is the upper arm bone of this animal. And this is what I have studied first at the university in Paris. And we were looking at the bone tissue of this animal to understand when they were getting adult and how they were developing and what was their biology and in which environment they were living. So it's a lot of information that you can actually get out of the bone tissue of these animals. And now the fossils I work on are slightly bigger actually, and I can share an image of the fossils I work on. Yeah, here it is. So at the top here, this is one of the fish I've been working on, which is called Eusthenopteron, and which comes from the Arctic Canada. And you have under one of the earliest tetrapods. So you have the tail of one of these earliest tetrapods. And here you have the hind limb. And so this is the reconstruction that was made in the 1930s with the head of the animal, the vertebral column, and the tail. So that's roughly the specimens I work on.
We will get into understand how you actually can get this information out of a stone.
Yes,
that's kind of. But I know that you work with X-rays, but how do one traditionally investigate these fossils, and why do you today use X-rays instead?
Yes, exactly. So you're right. Originally, like traditionally, we were actually cutting the fossils. So that means that you embed your bone in a resin, and then you use a saw, a very powerful saw, to section your bone. But the problem with this traditional technique is that you have to damage your sample, which means that once you have sectioned your bone, you have lost part of the sample, and then you can't use it for like other type of studies, morphological studies or biomechanical studies. So that has been quite a big problem to study this specimen coming from Greenland, because they are so precious. It's so hard to actually be able to go on the field, to do some research, collect the material, bring them back. So because of all this logistic problem and the fact that these fossils are unique in the world, we have, like, for example, 4 only humeri of one specific tetrapod. And sometimes we only have one bone of this tetrapod. So any curator would just not allow you to section it. So we have been developing, and this is Paul Taforo at the European Synchrotron Radiation Facility, who has been developing this technique. So it's a technique to image in 3 dimensions the bone. So you get some information of the inner structure of the bone without damaging the sample. And that's quite important, like, and crucial for our research now.
So basically you take this that you showed us, this fossil, and you put it under the X-ray instead of cutting it up. So then does this mean that several researchers can investigate the same fossil now, or do paleontologists keep on to their fossils? Do you have to go out into the field and find your own, or how does it work today in the field?
Yeah, so we are all shared a bit with specialties. So there are like people going on the field and, excavating more and more to find more fossils, because the fossil record is still quite lacking some information. So we always need to get more and more fossil to understand our past history, evolutionary history. But as well, you have also some researcher who actually collect the samples in the collections, in the museums. And so from these samples, you can go and do some 3D histology studies on these samples. So usually when you publish the data, you also have to make the data public. So that means that once you have scanned your sample, everyone can also use the data to study other aspects or check out like what you have done.
I would like us to dive in to really understand how you can extract information from the X-rays of these fossils. I know you have a movie to show us. Can you talk us through how it looks when you are investigating these fossils?
Yes, I can do that. So I will share my screen again and then show you the movie. Yeah. So can you see it well? Yes, right. Okay, so then I run the movie. So here you have the upper arm bone, so the humerus of a 380 million years old fish, so the Eusthenopteron fish I was showing you. And here, this is a high-res scan at the submicron resolution of a tiny piece of the cortex of this bone. And so you're traveling inside the trabeculae, so these are all these peaks, and now you're going in the cortex, and in the cortex, what you can see here, these are all the spaces for the blood vessels. So from this 3D imaging, you actually get some information about the blood vessels, the cell spaces as well, because this is very important for understanding the physiology of the animal, and as well the organization of the inner structure that we call the trabeculae.
So what does this information tell you about these animals, and what kind of conclusions can we draw from this information about how we moved from living in the water to emerging up to land?
Yes, exactly. So the bone is actually a bit like a book. When you open it, you get all the information from the first day of the skeletal development of the animal to the last day when they were when they died, actually. So you can very much follow all these layers of bones which have been deposited along the development of the animal. And it's a bit like, so I used to show that to my students when I'm teaching, but it's a bit like a tree. So this is a branch tree, which has been sectioned. And you know, this is the beginning of the tree when the tree was very, very little. And then when it started growing, you can see all these cycles, all these rings. And this is exactly the same in the bone. The bone will start as a small ring originally around the cartilage and will start like growing, depositing more and more. And while it's depositing, it will incorporate blood cells, blood vessels and bone cells. And all this information will give you some clues about how fast the animal was growing or how slow it was growing. So for example, if you compare like a tiny, let's say amphibian, and if you compare a dinosaur, you will get a lot of information out of the blood cells and the blood vessels, as well as the bone cells. And I can share as well some images that I have to illustrate a bit that in 3 dimensions. So again, this is the fish I was talking about, Justenoctoran. This is the upper arm bone that you have seen here. And you can see that you see in blue all the spaces for the cells. So if the cells are big, if the cells are numerous, you can know that the animal was growing fast. And you see as well here in pink, all the spaces for the blood vessels.
Let's see, we don't see the pink now, we only see the blue, really. Oh, there.
And here, yes, pink, there, okay. Red, actually here behind, hidden behind the cells, like in between the cells. And this is the spaces where you have the blood vessels. So if these blood vessels are also numerous and big and have a big volume, that means that you needed a lot of nutrients, you needed a lot of blood cells, blood elements, oxygen to actually make the bone grow very fast. So all this information is crucial to understand how the animal was growing fast or developing. And if they could reach the sexual maturity, they could become adult early in their development or later in their development. And there is another aspect, which I will show you here, and which is inside the marrow cavity. So inside your bone, you have a hollow space. And in this hollow space, you have marrow cells. And these marrow cells, they are very important because they will produce the blood cells. And the blood cells, like there are millions of blood cells produced every day in humans. And these blood cells, they are produced inside the marrow cells. And in this 380 million year old fish, we were able actually to see just the premises of the tubes in which you had the marrow cells forming. So we didn't have yet the production of blood cells. This happened after the water-to-land transition, but at least in this fish, you have the first, architecture of a real long bone that we have in our limbs and legs and arms.
How unique is this data? Are every scientist doing this, or how does it work? I know that you go down to Grenoble, to the synchrotron in Grenoble. So how does it work? How do you work to get all of this? And is this something everyone is doing today, or is it like the state of the art, what we can do today?
Yeah, so actually accessing a synchrotron is quite difficult because you have to go through a process where you apply with a proposal, and then you have a selection of these proposals. And in the imaging beam lines, it's quite like the selection is quite high because it's something which is very visual, so very helpful for the research of a lot of researchers in biology, in geology, in paleontology. So it's quite competitive, I have to say. But once you get access to this synchrotron, the synchrotron is a bit like a medical scanner, but with much more power in the beam. The fossils we're investigating can be that big. So this is another bone. This is also a long bone. This is just the head, of one of these bones. And it's extremely big. Like if I compare it to the small, tiny sample I was showing, it's really, really huge. And using the synchrotron, you can get access either to this information or the other one. So that's the beauty of this, this machine, actually. So this is kind of a particle accelerator, okay? So you have electrons spinning around, and when they are deviated, they actually emit X-rays. And we use these X-rays to scan the sample. So it's as if you were going to the dentist and you would do a radiography of your teeth, but here the beam is much more powerful. And this is what allows us to go through and penetrate fossils which are that dense to get tiny details up to the sub-micron resolution. So it's an extremely powerful and state-of-the-art technique that we're using now. So it's not something that you can access easily. It's really, really like you really have to justify why you can't section your bone or damage your bone and why you would need this information in 3D, for example.
Mikael is asking here, does the X-ray influence the sample, destroy the sample?
Okay, that's a very good question, actually. So in these fossils, which are like between 380 and 200 million years old, there is nothing anymore that we can damage. They are like full stone. I mean, there is some samples of still organic matter, but it's not something we can quantify, for example, with DNA or proteins. But if you would like to scan like the skull of a hominid, for example, then that would be very important to make sure that you can use a secure protocol. And this is what they have developed at the European Synchrotron Radiation Facility in Grenoble. So they have a secure protocol that will not damage. So you will be able to image the skull, for example, or the teeth or whatever structure you want to image without breaking the DNA that remains inside, because you have also ancient DNA that you need to apply to these samples. So they have very specific protocols. But when you apply this protocol, that means you need more time, beam time to do the scanning. So definitely this is a constraint. And this is something they take care of at the European Synchrotron Radiation Facility. But for my fossils, I'm quite secure with that. Yeah, they are far too old.
So when you have had your X-ray or you have been in Grenoble and you get the information, the data files, which I suppose it is, and you start to image up, is the work done then, or what happens then?
Yes. So when you get, what you get radiographies out of your data, but then you have to translate your radiographies into teeth images. And then maybe if there was like a dust on the camera, or you want to remove all the noise that can be happening during the scanning session. So you will have all the process, which is done actually in collaboration with the local people, like the researcher, which are fundamental at the SRF and us, together to remove all this. And then after we get the teeth images, and then this is from our side, a matter of reconstructing a stack of teeth images into a 3D image. Or into a 3D movie, as you've seen just before. And that can take maybe several weeks, but that can take also maybe 7 years. It all depends on the contrast of your data, and it all depends on how much you can automate the data segmentation. We call it segmentation when you model that in 3 dimensions. So it's a long process. It can be a very long process, yes.
So I need to ask, you were talking about becoming the doctor of dinosaurs. So you have looked a lot of fish, but have you been scanning any other fossils also? Have you looked at dinosaurs?
Yes, yes, exactly. We've looked at dinosaurs. We've actually scanned the fossils of Archaeopteryx with one of my colleagues, Denis Voeten. So we have been working on dinosaurs, and so that was for that topic. That was the origin of the flight and how, like, Archaeopteryx, which is this iconic dinosaurs with feathers, was it able to fly or not, and how was it able to fly? So we look at the inner structure inside, and you get some information about how light the skeleton was it able to fly or not, how adapted to the flight it was. So yes, we've been working on dinosaurs bones. I'm like essentially focusing on the fish to tetrapod transition and the water to land transition. But we have been as well with some colleagues from Australia working on fossils which are even older, well, from the Devonian, so 380 million years old. And these were the first jaw vertebrates. And what we were able to do with these fossils, so they are coming from the Gogo Formation, which is in the north of Australia, and they are in nodules. So it's kind of like a big pebble, a big stone, okay? Very, very dense. And in this formation, the conditions were very specific. There were a lot of bacteria. The environment was a bit anoxic, so not so much oxygen, a lot of sulfur. And in this condition, when an animal would die and go at the bottom of the water, bacteria, a film of bacteria would straight come to it and protect it actually from any damage. So in this environment, you have specific conditions that make that even soft tissues were preserved in fossils. And it has been quite crucial. So one of our colleagues, Kate Trinastic from Curtin University, find out, like, so they were originally, removing all the stone with acid. They were just getting the skeleton. And once she realized that actually around the skeleton, there were, some remnants of structures. It was a bit dark here, a bit brighter here. And she realized that it was something that she could see in all the specimens that she had investigated. So she thought, maybe we have something else. It can't just be like random. We have something which is associated to the skeleton. And what else could it be except soft tissues? So she came to the synchrotron, to the European Synchrotron Radiation Facility in Grenoble, and she collaborated with Per Alberg and Vincent Dupré and myself. And we scanned the samples at the synchrotron all together, and we're investigating the soft tissues. And we were able actually to realize the 3D virtual dissection of all the soft tissues that you could see in these samples. So what could we see? We could see muscles. We could see that it had a small heart, and I will show you a movie afterwards. It had a very big liver with 2 lobes, a bit like in chondrichthyans, a bit like in sharks, which this oily liver that allows the buoyancy of the fish in the water. And so we could get the first 3D dissection out of it in the oldest jaw vertebrates. So now I think I can share a bit with you that. Yeah. So I just need to get to the movie, sorry. So here, this is a nodule, like this big pebble, very, very dense, no way to penetrate it except with X-rays. And then you virtually remove the nodule. And what you get inside in gray, this is the bones. So the skeleton of the fish. So this is an armor fish. So it had all this dermal bone on the top. And here, what you can see precisely, this is a heart. So, and with the arterious element at the top. And so they found out that the heart was made in 2 chambers, the atrium and the ventricle. And it was very, very anterior, as in bony fish. So you have some shared character between the sharks and the bony fish like us. And this is what made this study very important, because this is the earliest steps of our evolutionary history And that was really, really neat to see all these details about the tissues and the structure of these animals.
So this is actually one of the first time we really could investigate soft tissue in a fossil.
So no, there have been other studies made also with the synchrotron to investigate soft tissues. So in a chimera, so which is another type of shark, they were able to see the brain in the brain case of a fish as well. So that was also a very big studies, because it could also allow us to understand how the brain was evolving. And we figure out that the brain was actually very tiny compared to the brain case. So, you know, you would suspect that the brain would just take the entire space, but it wasn't the case. And there was also a heart study made in a more recent fish that was also imaged at the European Synchrotron Radiation Facility. So now we are trying to investigate soft tissues whenever we can. That's really exciting.
I can understand. We have two questions here. Malin asks, are these types of fish fossils hard to find?
Yes, in these conditions, like with soft tissue preservation, yes, it can be very hard because you really need some specific conditions of preservation. So it can be very hard. But we are trying to get more and more of them. And now when we see, you know, some remnants of something in the rock, then we just go to the synchrotron and we check out if there are soft tissues or not. So it's getting more and more common. And thanks to this new method, yes, definitely.
How long time, Mikael asks here, how long time in the synchrotron for one such measurement? So
how long does it take to do the scan, the actual scanning session? It's usually like 3-4 days. So in like a nodule, for example, of this fish could be imaged in, let's say, like a few hours. So we do scan like several nodules. And if you want to go at higher resolution, then you need to change the setup and do a scan at higher resolution. So I would say like usually the scanning session for like the study on the oldest vertebrate was 4 days in total. So in 4 days, we could have like enough material to investigate and segment in 3D for the study we had. Yeah.
So let's go back to the transition from water to land. The work you have done in the synchrotron, looking at skeletons, understanding the soft tissue and the development of this species. What do we know today about this transition?
Yeah. So for example, about the marrow processes I showed you, you know, these tubes that we were seeing in Houston Afternoon in 3D. This, we can know now that actually the earliest tetrapods were not having like blood cells produced in the marrow as we were suspecting, but this probably happened later when the animals were moving much more on land. So this is something we are continuing to investigate. As well, we were able to investigate the life history trait of this animal. So when I speak of the life history trait, this means, you know, the development, how fast they were growing and how long they would take to become adult. And we could see that actually they were very, very slow animal in their growing aspect. I mean, they were becoming like adult at around 10 years old, which is really something long in the realm of animals. So we tend to know more and more about these animals and in more details about their biology.
So we are approaching half an hour now, and before I end this session with you, I would like to know, where are you going? What is your most inspiring scientific adventure you can go on? Where do you want to be in the future?
So we definitely would like to continue with this soft tissue anatomy. And because this is what we are missing in paleontology, we usually focus everything on the skeleton. And this is how we make the relationship between the animals. But now soft tissues are getting more and more important. And so we want to develop new techniques to actually really map the muscle insertions on the bones and really see how this will be evolving in the earlier stages and later on. So that's my main target now.
Thank you, Sophie. next week.
Thank you.
AppTalk Weekly will be in Swedish, and we will meet Jordi Luis Figueras in a conversation about one of the most important numbers in mathematics, P. Together with Jordi Luis, we will address what P is, how it came about, and why the number has gained so great importance in mathematical contexts. With this, Sophie and I wish you all a good week, and thank you for joining us today.